This is Audiommunity, a podcast about our body's never-ending fight with the outside world.
Hi everyone, this is Audiommunity, recorded on July 25th, 2018, and it's been some time since we recorded, and longer since we released an episode, but I'm sorry about that. I'm Kevin Bonham, and I'm joined this evening by Chadene Tremeglio.
Hello.
And Camilla Engblom.
Hi.
And Matt and Kate are stuck doing other things. The summer has just been really hard to schedule. I don't know.
Yes.
Like, people have been traveling.
Yes.
I've been to Europe twice.
Ooh, just rub it in, Kevin.
I just,
Look at you.
It was for work. It wasn't like super great time. I just got back from a 7-day meditation retreat.
Yeah, you were silent meditation, right?
Yeah.
Oh my God.
So you have a lot of talking to make up for.
I recorded a podcast during my silent meditation retreat.
That would have been ironic.
Quite interesting. What beer are you drinking, Kevin?
This evening I'm drinking Whirlpool from Night Shift Brewing, and they call it a New England Pale Ale.
Ooh.
I don't actually know what that means. There's a New England IPA style, and I don't know exactly how this fits in, but it's just a little bit hoppy, it's a little bit complex, and quite tasty. And a nice tall can, which I need because it's friggin' hot today.
It is muggy and gross.
It's really warm. I was telling you guys, I had to blast the AC before coming here, or before starting to record, because you can't have AC and record. At least not my AC, because it sounds like a spaceship is taking off. But so I just blasted it, and now I'm hoping that the cool will stay inside the room for the extent of this.
Well, good luck with that. Or hopefully it will warm up slowly enough that you won't notice.
Oh, yes.
I just biked home and then came immediately to record, so I am dripping sweat, which I'm sure you all wanted to know.
Very nice.
Camilla, what are you drinking in your nicely chill room?
Well, I'm drinking another beer that I found in my fridge, which is from the Aeronaut Brewery in Somerville. And this is a higher quality than the beer I usually find in my fridge. And it's an IPA, and it says "A Year with Dr. Nandu" on the side.
Is that the name of the beer?
It's called Nandu.
Okay.
And then it says "A Year with Dr. Nandu." And I don't know, there must be a story behind this. It has a really cool design. There's like a hot air balloon and a strong guy on top of the U in the Nandu, and then a bunch of squirrels and an airplane that looks like a fish.
The hot air balloon, I think, is the sort of logo of
Aeronaut
Brewing. And their brewery is really awesome. If you ever get a chance to visit, it seems we're both drinking local brews. Night Shift, the beer that I'm drinking is... their brewery is actually right next to the climbing gym that I go to up in Everett.
I think I've been there once. Have I been there?
It's great.
Yeah.
Their beers are a little pricey, but they're quite tasty. And Chadene, I won't ask you because you told us ahead of time that you're just drinking water.
Yes, a very complex, lovely, hoppy glass of water. No, seriously, my summer has been really stressful for many reasons, and I'm pretty sure I gave myself an ulcer. So I am taking Zantac and drinking a lot of water.
Wow.
Deeply unpleasant.
That sucks. I'm sorry to hear that.
Yeah. I'm okay. It's mostly, it's like just I'm getting old and I can't eat the kind of hot, spicy food I used to love, you know? It's just killing me.
Getting old.
I am, though. Seriously.
No. But it seems like you have had a busy summer.
Yeah, it's been crazy here. It has been nuts. Kids home in the summertime you'd think it's like calm because school's out, but that's just when all the activities pick up and the play dates and the craziness. So it's been busy.
Wow.
Well, okay, let's find a transition. It's been busy.
Kids, osteoblasts.
So now we finally got time set aside, at least the three of us, to return to an episode that was recorded like 4 months ago, 6 months ago.
Long time ago.
I don't recall exactly, but we had
I want to say it
was
December.
Oh, God.
This was recorded. I'm really happy. Well, I'm a little happy that we get to redo it because it's Camilla's paper from her PhD. And I wasn't there the first time you recorded.
Oh, that's right.
I think
it was actually the reverse. I think so. Matt was there, and I don't know if Kate was there.
No, anyway, it was just the three of us. So Matt was there.
So I'm taking the place of Matt today. I did read the paper back then, but have not read it since. So this is going to be like seeing it for the first time.
But it's
I know
good.
It's about bone.
Yeah, it hasn't changed since then, so that's good.
Right.
Oh, perfect.
No retractions, no revisions. That's excellent.
No, and it's and I want to say it's my fault that this re-recording happened, because my audio as it was being exported got chopped into little 6-second bits and rearranged in no apparent order. So trying to paste it together was an absolute nightmare. So these kind co-hosts have agreed to re-record this.
Yeah, I still don't really understand how that could have happened.
I think it had something to do with it being saved in Dropbox, and somehow it was syncing at the same time as it was saving, and then it just got confused, and then it just, because I think there are little snippets, but it knows, like, it saves a whole file that's a composite, and then it never got to that point.
Well,
wow.
I have to say that Camilla did attempt a heroic re-recording of just her part, listening to her previous audio, and then re-record, which was silly.
Extremely awkward.
I think I tried that like for episode 2 or 3 because something similar happened, and it's just impossible to do it and not make it sound ridiculous. So I appreciate the effort, but I definitely think it's worth this re-recording. So we should say the article, which is osteoblasts remotely supply lung tumors with cancer-promoting Siglec-F high neutrophils, in which there is a ton of jargon, which we will hopefully get to.
Break down.
And it looks like this was published in Science.
Yep.
Is that right?
Yep.
Oh, that's very impressive. I didn't even realize that. Published in Science. First author, Camilla Engblom, friend of the podcast. And why don't you say the last author? Because
this is my PhD advisor, Michael Pitet, and he is a PI at MGH and Harvard Medical School here in Boston. And I should say that another postdoc in the lab, Christina Fürske, was also a co-first author on this paper. So it's a joint endeavor. And then it turned into this huge collaborative effort between lots of different types of lab contributing their expertise.
Like 20 authors on here.
Yeah, they are. There's quite a crew, but there's a lot of people in the lab who contributed, at various points in time. And then also, because it turned into this sort of complex multi-organ conversation, it involved bone experts, and then single-cell RNA-seq experts, and then tumor experts. So it kind of grew and expanded during my time in grad school, which was a lot of fun.
Cool. Yeah. Seems like you probably learned a lot here.
Yes.
So maybe, I don't know if you have a particular way you wanted to walk through this, but I thought it might be useful to sort of step through the title, because I feel like there's a number of things that are not necessarily common terms when thinking in immunology. So obviously cancer promoting should be clear what that means. And we've talked about neutrophils, I think, a little bit on the podcast before. I think we did a podcast on neutrophil extracellular traps one time. But maybe you could tell us a little bit about what osteoblasts are, and then what Siglec-F is, and why they might promote cancer.
Yeah, so osteoblasts are these really cool cells, which are bone-forming cells. So in the bone marrow, the cells that produce the proteins and basically mineralize the bone matrix, so all the sort of hard content that makes up your bone, are produced by these cells called osteoblasts. And this is not to be confused with osteoclasts, who are aptly named almost exactly the same, which are the cells that break down the bone. So you have these osteoblasts that live in the bone and help produce bone, and then you have osteoclasts that actually break down the bone. And this is a continuous cycle, making more bone and breaking down bone that occurs all the time in your bones as you speak.
So this is actually somewhat, I find it a little bit surprising, actually. I feel like I may have learned this. There was some other immunology grad student that gave a talk at a retreat in our program one year, but I sort of had this impression that bones grow as you grow, and then by the time you sort of finish, growing in puberty, that your bones were sort of like set.
Right. So I think they definitely turn over less frequently after you've gone through puberty, but there's always some remodeling going on. So you always have osteoblasts laying down new bone together with osteocytes, which are actually even cooler cells that live inside the bone. And they have these huge matrices that dendrites basically, that stretch out from the osteocytes and they touch each other in the bone. But you constantly have new bone being formed and the osteoclast breaking down the bone. And then, so when women go through menopause, for example, they lose a lot of estrogen, which is something that is needed for bone production. And it basically skews this turnover to increase the breakdown. So suddenly you have more breakdown than you have bone formation. And so then you start losing bone more rapidly.
Osteoporosis, right?
Yes, osteoporosis. So you have this balance that's continuously going on, but you don't really notice it because it's kind of status quo. And I think the estimate, I think this is in an adult, that it takes about 7 years to completely replenish your whole bone tissue.
Oh, wow.
So it does, it's not fast, it's not like your gut or your skin, but it does, slowly turn over time.
So I guess, yeah, it's interesting. We sort of associate the skeleton with like being dead because it gets left over and takes much longer to decompose.
Yeah.
But bones are actually living tissue.
Exactly.
And have a lot of the same properties, or at least,
And people are finding out that they're doing a lot more than just making bone. And so I think that's one of the hottest topics, I guess, in the bone marrow field in the past few years, maybe past decade or so, that people started discovering that it's not just that these osteoblasts make bone. They also have other functions that are related to sort of metabolism or even some neuronal symptoms like feeling hunger or things like that. So there are new things coming out all the time that, bone as an endocrine organ and not just as the structural matrix that holds your organs in place.
That's what I think is so cool about your paper, Camilla, is, this idea that bone has some sort of systemic involvement in a distant cancer. That is such a neat concept.
Yeah, I think it kind of goes back to this idea that cancer is not a localized phenomenon. And this is something that our lab has thought of quite a bit, that, you have a solid tumor. So we mostly focus on solid tumors, but you have a local tumor and it affects, the cells and the tissue in the area that the tumor is formed. But then you also have effects that go beyond this local tumor area. And these effects can act on the immune system. It can call cells to come to the tumor, but it can also act on other tissues. And it's really here where this project started, because we were thinking about, okay, we have all these different immune cells in the tumor and they come in different flavors, but most of these immune cells are not actually made at the tumor site. So they're produced in a distant tissue and they travel through the blood and they get called to the tumor and differentiate and change and can both influence, make the tumor grow faster or inhibit the tumor from growing more. So it's kind of this interplay, like what happens at the tumor site doesn't really stay at the tumor site, it goes beyond that. And what are the different tissues involved? And that's kind of what took us to the bone marrow and to start this project.
Right. So you're looking at lung tumors, but there's something happening in the bone. And maybe before getting into the paper, is there sort of a primer on what cyclic F high, were cyclic F high neutrophils something that was known before, or what is cyclic F and how does it relate?
Yeah. So basically cyclic F is a receptor and it binds sialic acid. So this is basically a motif or a protein modification that basically is a sugar modification that binds to the cyclic F receptor. And the cyclic F positive neutrophils were not known before. So basically, cyclic F, if you're an immunologist, which I know you are, Kevin, but for the
at least it was at one point.
You still are. And then once an immunologist, always an immunologist.
Okay, that's good to know. Thanks.
You can never leave.
My immunologist card.
No, so it's been associated in mice mostly as being on eosinophils, which is another type of immune cell mostly involved in allergy, or it's mostly infamous for its roles in allergy, I would say, and also sort of helminth infections. And basically, this is used as a marker for eosinophils in mice, but also other types of cells in the lung. Some macrophages in the lung also express sialic F. So basically, when we were looking at these tumors, I was tossing in, different antibodies, so different against different surface receptors to try to figure out, what are the different cells that are in this tissue and how are they changed. And I was really using this antibody to rule out that they weren't eosinophils. But it turns out they actually ended up being a subset of neutrophils that expressed this receptor only in the tumor and not in healthy tissue. And so for us, it was quite exciting because it was this first time we could use the surface receptor expression pattern to basically see that it was an immune cell that was present in a tumor-bearing lung. Okay, to say that differently, but basically, usually when you look at an immune cell and see what proteins are expressed, you can't tell whether that cell has come from a tumor, a tumor-bearing lung or a healthy lung. But with this type of receptor expression pattern, we can actually say, okay, well, this actually came from a tumor, a mouse that had these tumors in the lung. And so,
cool.
Yeah, sorry.
No, I was just going to say, so maybe we should start walking through the figures?
Absolutely. Let's walk.
Unless there is some other background that you think would be useful.
No, I think this is great. And so I think I won't go through every single panel because it's quite a lengthy paper. And I think we don't want to age significantly and also die of heat stroke in this non-AC room. But basically, the main gist of the first figure was that we wanted to know. So we had these tumors, and we had lots of immune cells in the tumor that come from the bone marrow. And we realized that there wasn't much known about what the bone was doing in the context of a cancer. So I mentioned that people had started figuring out that there were other functions of bone, but most of these studies were not done in the context of a solid tumor that was not in the bone. So we really wanted to see, okay, is anything happening to the bone when there's a tumor, at a different site than the bone? So we had mice with lung tumors, and we asked, is the bone changed?
Is it worth describing what the tumor model is?
Sure. So we used different types of tumor models, but the main genetic mouse model we used is a model developed by Tyler Jacks at MIT and people in his lab. And this is a pretty neat model because it's a genetically engineered mouse who, when you don't do anything to these mice, they're completely normal. But when they are infected with a virus that contains an enzyme called CRE, you basically chop up the DNA sequences in in the cells that receive this virus, and they start, they're genetically engineered so that they start expressing an oncogene, so a gene that basically turns tumors on. So basically initiates tumors. And it deletes another gene that's important for restricting tumor growth. So the two genes are KRAS, which is turned on, so an oncogenic KRAS. And this is a mutation that's usually found in patients. And this gene gets turned on when you give this virus. And when you, and then P53 gets turned off, which also often happens in patients. So it's basically a way to mimic, how tumors would form in a patient, because a lot of mouse models rely on transplanting cells, that you have grown in a tissue culture setting, and then you inject the cells into the mice. But in this case, you're actually initiating tumor growth inside a cell in the mouse, and so the tumor can kind of grow more naturally, I would say.
Nice.
Yeah, we also used sort of transplantable models in this paper, so it kind of goes back and forth, but that's the basis of that model. And so whenever it says plus add CRE, this is this virus with the CRE recombinase.
Cool.
Yeah. So I guess in the first figure, the main takeaway is that we measured bone in different ways. And we used a bone imaging approach called OsteoSense. And this is a cool tool. So you can inject this probe into mice, and it mimics some of the agents that are actually given to patients when you want to look at their bone turnover. And you inject this probe, and then you can measure this with fluorescence molecular tomography, and you can study, the more signal you have, the more active remodeling is going on. So the more remodeling of the bone. So you have a stronger signal that indicates that you have more sort of turnover of your bone. And that's what...
So this means, when you say turnover, do you mean a combination of osteoclasts degrading bone and osteoblasts building bone, or do you mean an abundance of one or the other?
Yeah, so that's an excellent question. And it's actually, this tool is marketed as a bone formation, as a measure of bone formation, so osteoblast activity. And it's a bisphosphonate derivative, so basically... it labels newly mineralized bone, but it doesn't really measure the amount of bone. So it's not directly, so it indicates that there's more bone being formed, but you can't, without measuring actual bone density, you can't really say that there's more bone, if that makes sense.
Okay.
Yeah.
So you don't have the osteoclast picture from this.
Right,
bone.
Exactly. So they could be like tons of new bone being formed, but you could also have tons of bone being broken down at the same time.
Right.
So that's a super nice segue into E, because A through D in this figure basically measures osteosense activity in different mouse models, and using both this FMT, so the fluorescence molecular tomography, where we measure this osteosense signal in live mice that either don't have or have tumors. So that's an A. And then we quantify that in B, and we use a different tumor model in C. And then in D, we look at it by microscopy. And
it looks like you're looking in, just to be clear, you're like, even though these are lung tumors, you're looking in the femurs of mice and their legs, basically.
Yes, exactly. And so one of the concerns we had was, okay, maybe we have tumors in the bone, because even if the tumors are in the lung, maybe they metastasize. So we did some other tests to show that, okay, we can't detect tumor metastasis to the bone. So
cool.
In all likelihood, the tumor is in the lung, and whatever is happening is this, this goes back to the title that's remote activation from one site to the other.
Right.
And so after we found, and the takeaway message here was really that, okay, we found increased activity or increased osteosense signal in mice with lung tumors. So then we said, well, we don't know if we had more bone or less bone, or we think it's more active, so probably more bone, but we want to know. And so then we used micro CT, but for mice. So basically looked at bone scans of these mice and measured the bone density. And we saw that there was actually more or higher bone density in the mice with tumors compared to the tumor-free.
So Camilla, I know I asked you this before, but was that a surprise to you guys? Because I guess, when I think of cancer, I think of wasting. Like I would have assumed maybe less bone. So was that like a surprising finding, or?
Yes, it was definitely a surprise. And I would say, that's the main association. And that's definitely true that a lot of cancer patients suffer from bone loss. And I think part of why it makes it part of that association is because a lot of the cancer treatments that people receive dramatically cause bone loss. So chemotherapy, for example, radiotherapy, and a lot of androgen deprivation therapies, so like hormonally disruptive therapies. So where you would block estrogen, for example, or block male hormones, you would basically cause bone loss, because that's basically what happens, like we mentioned with menopause. So but you're doing it because you want to stop the tumor from growing, but you're causing like early menopause in a lot of women who have breast cancer, and that also affects, you get the bad side effects of menopause as well, which are the bone loss components as well. So there's that association. And actually, when we started looking into this, we were really trying to find a study that had looked at people, before receiving any treatment for cancer. And that, you had a control group that did not have cancer, but otherwise were mostly healthy. And then also did not have cancer in the bone. And we couldn't really find any study that had addressed this point. Mostly it was like, you have cancer, and then we follow these people over time as they receive treatment, and then you see bone loss. So we were able to work with a clinician at MGH to look at a smaller cohort of patients. So we looked at 70 cancer patients in total, but looking at scans, so CT scans of their thoracic vertebrae. So these are the ones that basically are at the top of your spine. And so, these patients went in because they had, symptoms of lung cancer, and they received a chest scan because, you also scan your chest cavity. And basically then we could look at the bone in that area. But we only looked at patients that did not have bone metastasis. And then we could kind of match these patients to otherwise healthy individuals. As far as we know, right? That were both kind of age and sex matched and, didn't receive any treatments. And we also saw a similar phenotype there, that there was a small increase in bone density in the patients with lung tumors compared to the controls. So I think you definitely need to do more studies here. I think we have a first, cohort looking at this. And there have been I gave a talk at MGH, and one of the clinicians came up to me and she said, I've heard of a syndrome where, if someone comes in and they scan their arm, if they have, some bone pain or something like this, and they see like abnormally high levels of bone, then they immediately look and screen for lung cancer. So I was like, what? And this was after the paper was out. So I was like, okay, that's really cool. So there are I think there are a lot of things we don't know yet about this, and we need to figure out more about the human phenotypes here.
So, but I think this is really cool. Like, I think this is not done enough where you have in your first figure, you're like, here's an interesting observation in mice, and then immediately say, is this plausible? Like, is there a plausible link to humans?
Yeah.
And I really like that you include that in the very first figure. I think, a lot of times people will go through an entire paper and at the very end they'll sort of have and maybe this applies to humans. Yeah. But
thanks to reviewer 3.
Front, I think is right. Really nice, nice sort of justification for spending the time to go and do the rest of this stuff.
Yeah, I think, I would love to say that the story was as linear as it's presented here, but it's definitely, everything kind of happened in different order of things.
Yeah, of course.
But it was definitely, once we had the mouse data, we said, okay, we really, if we can, get human data, that would be amazing. And it was really, I was presenting to the Center for Skeletal Research at MGH, and they said, oh, well, we know someone who, and that's really how it happened. And it was really one of those moments where you're like, okay, it's Boston is an amazing, other places are amazing too, but it's really a neat place to be here where you can have an expert, right next door, and they can help you out with these kind of things.
And you should keep going. I'm going to step away and go grab another beer because it's so damn hot and I need more to drink. So,
Chadene, hold on the fort.
All right. I can do that. I can do that.
But keep going.
Yes. Kevin needs a beer. Okay. So for figure 2, what we wanted to, now that we kind of had an idea that something was happening in the bone, we wanted to say, okay, well, what's happening on a cellular basis? Sure. And here, Kevin mentioned that it's nice to have human data, and that's what you said that too. And yes, it's really really incredible to have human data, but it's also, to do anything mechanistically is almost impossible. So then we're like, okay, let's go back to the mouse and let's try to figure out if we can, think more about what happens on the cellular level. And all of figure 2 is really dedicated to looking at different ways to assess osteoblast activity.
Gotcha.
So it shows in figure 2A, we're doing it's a trichrome staining, which is basically a way to look at both bone and cells in the bone tissue. And here again, we're looking in the femur. So this is basically the top of your femur bone, which would be your knee joint here. Mm-hmm. And the turquoise part is basically the bone. And you can see, like, in the middle top panel of figure 2A, you have these cells that kind of stretchy and they lie on top of the bone. And these are osteoblasts, and that's what they look like when they're not laying down bone. So they're kind of stretchy, long, and flat.
And these are sort of inactive ones.
Exactly.
Okay.
Exactly. And when they're activated, and this is, a representative example, in the panel below, they get this cuboidal shape. So they kind of curl up. And then
big difference.
Yeah, they're kind of cool looking. They start putting down bone matrix. And actually, this staining is cool because you can see the new bone as this sort of bright purple stain that's on the turquoise. And so that's new bone that has just been formed. So it's like just mineralized. hot
That's
off the
cool.
osteoblast.
That is really cool. I love staining. This is a gorgeous image.
Yeah. No, there was an amazing lady called Janaina at the Center for Skeletal Research who did these, and she's just phenomenal. And she's also one of those people who's wonderful to work with. I mean, like, oh, can you please help me with this? I'll take care of it. Don't worry, Camilla. Don't worry. I was like, yes.
Nice.
So she was fantastic. And another thing that I think is what I like about this figure is figure 2E.
Yeah, take me through this one.
Yeah.
I'm a little, I need a walkthrough.
So this one is nice because it's a way, so it shows bone formation. And so the little arrow is basically showing the amount of bone that was formed over time. And so just to like break down, the picture again is of the femoral bone. And it's showing two lines, like one in red and one in green.
Right.
And these are basically fluorescent labels in the bone marking the time when these were injected. Because basically when you inject them, they get incorporated into the bone. And then as the bone grows, the marker moves into the bone. And then you inject again, and then you have another marker. And then you can measure the distance between the two to see how much new bone was formed.
Gotcha.
And so basically you have the mice, you inject them, you wait, about a week, and then you inject again with another color, and then you take the bone, you section, and then you put it under the microscope, and you have these, fluorescent tags for when you injected.
Wow.
What is the hash mark doing there?
So the hash mark is telling you that this is the bone side, and so the other side is the bone marrow. So you see all these round little cells next to the red.
Sure.
So it's basically just to orient ourselves in the tissue.
Got it.
But I think...
And I have to say, like, Chadene mentioned that she really likes the histology. I've always found histology to be really opaque. I mean, these ones happen to be, like, super clear because the stretchy ones versus the ovoid ones are really obviously different. But I really love the quantitation that you're doing. So there's for the rounding of the osteoblast, you have number of osteoblasts divided by bone surface, or just the osteoblasts that are touching the bone surface? Yeah,
so it's osteoblast over the bone surface that is increased.
And then for this last assay you were talking about, you've just got basically a straightforward measure of the distance traveled over the same period of time, and like the quantitation is really impressive.
Yeah, agreed.
And again, like this, Janaina was amazing with this because she, like, to be able to do this is not something you learn over one day. Like it takes years to really become really good at saying, okay, this is an osteoblast and this is this type of cell and this is, and we did some of it together, but for E, like she was really fantastic in this because it's basically... You have to know exactly what areas to measure and the way you convert, this distance into like actual minerals being laid down, so they can, based on, the type of measurements that you do, you can convert it to, like, it actually becomes a measurement of how much material these osteoblasts are putting down into the bone. So it becomes, not just that there are more of them, but that they're actually, actively laying down bone.
And this is Janaina da Silva Martins, who's the fourth author?
That's correct.
Right? Okay.
Yeah, super bone expert. And so then we were at the point where we said, okay, we have more bone in the mice with tumors, and we have more osteoblasts, suggesting that these osteoblasts were doing something different when there was a lung tumor. And at that point, we became very interested in saying, well, we have this phenotype, we even see it in people, but is it really important? Does it matter to how the tumor grows? And, do we care about this phenotype at all? Because ultimately, we want to know, can we figure out things that change how the tumor grows to, potentially find a new way to limit how tumor grows or figure out basic things about how tumors grow. So,
right, because it could be an effect, like the tumor is just producing some downstream effect that is irrelevant.
Exactly. And you say, well, you have more bone. Okay, well, great. Why do we care about this? And so here, now that we could kind of zoom in on the osteoblast, we're able to have a model where we can deplete osteoblasts in the mice. So we could ask, if we remove these cells... Oh, there's a lot of fuzzy sound suddenly. Yep. Yeah, now it's better. And
your paper is cursed, Camilla.
I don't know what is going on. I'll have some more beer. It sounds like there's a tornado inside.
Yeah, it just vanished.
Yeah, gone.
And we can't hear Chadene either. So
Did you?
This is going to be fun to edit.
Sorry, Kevin. We'll quickly get through the end of this paper.
No, I mean it'll actually be pretty easy because it's just a matter of
it's just like one
taking
chunk
her audio and just chopping it out. This is why we record everybody individually.
Yeah.
I will say that is a
oh my God,
critical component
it
of
is
this.
so warm in here. I'm not made for this kind of weather.
I'm sorry.
I need
because you're from the frozen north.
Yeah, I think so. I need I walk around with like a wet towel on my head looking like a complete crazy loon, but I just...
Well, maybe while we're waiting for Chadene, I want to update listeners and let them know that I have started a new beer because I wouldn't want them thinking that I'm still drinking the Whirlpool. So now I am drinking a Conehead IPA from Zero Gravity Brewing Company that's based in Burlington, Vermont.
Fantastic.
There are a lot of really good breweries in Vermont.
Yeah.
It's a tiny little state, but they have a crap ton of great breweries.
Yeah, I feel like we need some stats on breweries per capita.
That would be interesting. I'm sure that exists somewhere.
Yes.
Because, well, I know, so I have my wife has friends from college actually that started a brewery in in Brattleboro, Vermont, called Hermit Thrush Brewing. They make like amazing sour beers. And there's they have all of this promotional material that just talks about how many breweries are in Vermont.
It's the place to be for beer lovers.
But maybe, because we're still waiting for Chadene. Oh God, her Audacity crashed. Oh, thank God. Apparently the file was saved. While we're waiting for that, I feel like we should just keep a bunch of this in.
This is like post-traumatic Audacity disorder.
Thank our Patreon supporters, because we forgot to do that up top. And at some point, I think we're going to have to buy Chadene a new computer, because hers just sucks. And that's going to be made possible by our Patreon supporters.
She's texting you right now saying, haha, thanks, Kev.
Yeah. No, we can't afford to buy Chadene a new computer based on our Patreon support. But if more of our listeners contribute 50 cents or $1 per episode, then we can at least afford to pay for the beer that the hosts drink on each episode. And I've said in the past, this is motivation for us to continue to record. And that might seem a bit hollow, given that we haven't actually posted an episode in 2 or 3 months. But I promise you that if it weren't for your support, it would be less likely than we record at any given time. So please, if you enjoy the podcast that you're hearing, just to sort of give us, it makes us feel good, and it will pay for the audio software that I use to edit these podcasts, so that I can remove all of the terrible sound that Chadene's microphone just made. And we just really appreciate it, and it's super motivating for us to have a base of support that's coming in every month or every episode that we actually post. So if we don't post in a month, we don't actually get the money, which, is good accountability for us as well. Another way that you can support the podcast is by sharing it with your friends. If you are listening, there's a good chance that you're a scientist that spends a lot of time at the bench pipetting, or in tissue culture hoods, or in mouse facilities where you're listening to podcasts because you can do that while you're doing your work. Mm-hmm. And chances are some other people that are in a similar situation. So if you think that they might enjoy Audiommunity, you should share that with them. All right. So, dear listener, I have just cut out probably about 20 minutes of audio in which one of our hosts, I won't name names,
my bad,
had a giant crash of audio software. We hope the previous audio was saved. If not, I'll put in a little announcement, because we are definitely not recording this podcast again.
It is saved. I promise you. I have it on my desktop. I'm looking at it as we speak.
I think where we left off, if I'm not mistaken, is that, let me pull up the paper again. We had just learned that bone is being formed, and the question is, do we care?
It's the scientific cliffhanger of autoimmunity. figure 3 comes to help us answer this question. So we wanted to know, do we really care? Is this important? And what's nice is that other people had already cared about osteoblasts before us. So they were models in which you could specifically delete osteoblasts and get rid of them. And you can compare these mice that had lower numbers of osteoblasts to those who had normal numbers of osteoblasts and ask, are the cells really important? Are they doing anything? So these mice, if we go into a bit more detail, they are they're also using the Cre enzyme that I mentioned previously. So in these mice, the osteocalcin promoter, so osteocalcin is expressed by mature osteoblasts, and the promoter for this gene drives the expression of the Cre recombinase. And so when you have this Cre, you chop out sequences of the DNA, which leads to the expression of the diphtheria toxin receptor. And mice don't usually have this diphtheria toxin receptor. It's actually it's a monkey diphtheria toxin receptor, and it's very sensitive to diphtheria toxin. So when you give these mice diphtheria toxin, the cells that express this receptor will actually die. And so this gives you a way to specifically delete the cells or get rid of them that express this DTR receptor. And
I remember when I learned about this idea, because this is a model that has been used to delete a lot of cells, and I remember thinking it was like sorcery. Like, this is one of the ways that biologists are like gods, that we can go in and just turn on this protein, and we give diphtheria toxin, which is totally harmless to normal mice, and it will just completely obliterate specific cells that we have chosen.
Crazy.
That's just magic.
Yeah. It's pretty crazy. And
also the word diphtheria is like ridiculously hard to say.
It is. I agree.
That's
why we say DT.
TH, one right after the other. And I'm pretty sure that my wife would tell me and have particular, there's like a word for that kind of consonant.
Oh.
I forget what it is.
It's a diphthong.
Like an unvoiced fricative or something. No, it's not a diphthong. Diphthong is like 2 vowels in a row. No, I think it's an unvoiced fricative.
Fricative? That sounds like sounds like a curse.
Which is what this episode is
right now while Camilla continues.
Fricative. Oh, fricative. Hence the reason we're scientists and not English majors.
Yeah, all I heard was thong, so that's my... no, I'm just kidding.
A fricative denotes a type of consonant made by the friction of breath in a narrow opening, producing a turbulent airflow.
Interesting.
Wow. A fricative consonant, e.g. F and TH.
I feel like this is what was happening in the microphone before, Chadene. It was a tight airflow and friction and...
Compared to a voiced fricative, I just want to point out, which is something like V. Like a V sound.
Well, that sounded really interesting. Do that again. That had a great, like...
What's going on over here?
It just has like great resonance.
Neutrophils, cancer, lung tumors, bone.
Woo.
Wait, so real quick before you jump into it, let me just ask you, how do these mice do when you knock out osteoblasts? Are there big effects on their health and well-being?
Yeah, you don't want to do it for too long. So we had kind of a short depletion scheme because we wanted the mice to, not do so poorly. But yeah, if you keep doing it for a long time, then they get a little bit, they do a little bit poorly. They lose some weight. They, get a little hunched over. But we kept the treatment pretty short so that they wouldn't get sick.
Got it.
More sick than the lung tumors that they had. So in these mice, we had the mice where we could deplete the osteoblasts. So in the figure, they're shown in green. And we had the control mice. And so because we're giving these mice diphtheria toxin, we wanted to control for the fact that diphtheria toxin itself might do something to the tumor. So to control for that, we used mice that did not have the Cre transgene, or they lacked the DTR transgene. So basically, in these mice, you could not deplete osteoblasts even if you gave diphtheria toxin. So those were our one control group. And then we also had the OCN Cre DTR mice that we did not give diphtheria toxin to. And what we found was that if these mice had tumors and we depleted osteoblasts, we actually reduced tumor burden in the lung. So by getting rid of a cell population in the bone marrow, we reduced the tumor burden in the lung, so at a distant site. So this goes. Yeah, please.
Are each of those dots an individual mouse?
Yes, sir.
Oh,
That's a lot of
wow.
mice.
Yes. This was 4 experiments, I believe, and a lot of mice.
Wow.
Yeah.
Is there a reason that you had to use such high numbers?
No, it's just we repeated the experiment many times, and so we just ended up pooling all of the experiments at the end because we wanted to basically, first convince ourselves that it really was true, and also, just include all the data that we actually had produced. And
cool.
Yeah, it was a long and arduous PhD project. It's both for me and the mice. So, yeah.
Well, probably longer and more arduous for the mice.
I would argue.
If we're being honest.
I would argue the same. Yes. Yeah, I think that's a challenging thing as a researcher, like to, especially in immunology and cancer research where you work with mice, you constantly have to think about this and try to be the best and the kindest you can be while, doing good science at the same time. So in this, using this model, so basically A and B just looks at tumor burden. So it's just different ways of looking at tumor burden. A is looking at lung weight, which we find correlate with tumor burden. And then B, we look at histology. So we actually, circle the areas where we had tumor and see that the tumor area is decreased when we deplete osteoblasts. And so now, this kind of goes back to the title where we said, okay, osteoblasts remotely control how the tumor grows in the lung. Because we started thinking about, okay, well, if manipulating these cells in the bone marrow has an effect on a tumor in the lung, what's happening in between? We don't think that these cells are traveling from the bone marrow. We think that there must be some intermediate. And like I mentioned before, immune cells are actually produced in the bone marrow and travel in the blood and arrive at the tumor site. So it's logical to hypothesize that, these may be the intermediate mediator.
Yeah.
And so in this figure, I'm not showing all the data, but we have supplementary figures for those who are really interested. But we looked at a lot of different immune cell types. And in this figure, we show the myeloid cells. And we found that the most striking phenotype that we had was we had lower numbers of neutrophils in the tumors where we depleted, in the mice where we depleted osteoblasts. So our hypothesis then turned into, we actually know that neutrophils can promote tumor growth in other models. And so we said, okay, maybe the osteoblasts are helping, promote these neutrophils to go to the tumor. And then at the tumor, they're helping the tumor to grow. And D basically shows that if we target the neutrophils specifically with an antibody that causes them to die, we reduce tumor burden in the lung. So the neutrophils actually have a tumor promoting ability in themselves. And I think I'll breeze through E, but basically it's just showing that we actually had lower numbers of neutrophils in the blood if we depleted osteoblasts, suggesting that, we're really controlling the production of these neutrophils that then arrive at the tumor. But an experiment that we did in F was using a technique called parabiosis. And I don't know, Kevin, if you've covered parabiosis before.
To be honest, I don't recall if we have, but I kind of think we have. But basically, this is another one of those biologists playing God things. But basically, you attach 2 mice together such that they share a circulatory system. So you can basically connect 2 mice and then see if changes in one transfer to the other. And it's it's a little bit crazy. You're basically making Franken-mice.
Yeah.
And so they share a circulatory system, but what we call the stromal cells, the non-bone marrow derived cells, are basically being kept in each individual mouse.
Exactly.
Yeah?
Exactly. So then we could ask, if our hypothesis is true that, these osteoblasts remotely control the lung tumor, if we provide a normal bone marrow to these osteoblast depleted mice, we should be able to kind of restore the neutrophils and restore the tumor burden in these mice just by joining the circulation. And so that's actually what we found. So we saw that in the mice where we had depleted the osteoblasts, if we put tumors in these mice, but then join them to control mice that were completely normal otherwise, we actually increased neutrophils and tumor burden in the osteoblast depleted mouse. So that showed us that basically something was traveling between the bone marrow and the tumor and mediating this effect. And figure 4 then is where we finally get into the cyclic F part of the title. So here we did this experiment that I mentioned to you before, is we started saying, okay, well, the neutrophils are interesting, but we don't know so much about the neutrophils. Can we learn more about what they look like and what they're doing? And so what we did was we started profiling the neutrophils more thoroughly. So what we discovered in panel figure 4A was that in tumor-bearing mice, we had a particular type of neutrophils that we couldn't find in the healthy mouse. So this is showing you flow cytometry data. So this is basically looking at the surface expression of 2 different markers. And Ly6G is on the x-axis and cyclic F is on the y-axis. And Ly6G is a well-known marker for neutrophils in mice. So actually neutrophils don't express Ly6G, but mouse neutrophils do. And so when you look for neutrophils basically express this marker. And we found that in the tumor-bearing scenario, so mice with these lung tumors actually had cyclic F expression as well. So there was a subset of neutrophils that expressed both Ly6G and cyclic F. And then, we characterized these cells in different ways and found that basically, to all intents and purposes, they look and smell like neutrophils more than any other cell type.
You actually have a quantification of the smell of neutrophils?
Yes, it's neutral, actually, surprisingly. Sorry.
I thought I was going to trip you up with that, but you managed to roll with it. I'm impressed.
I blame it on the beer, I would say. So in the rest of this figure, basically, the goal was to investigate whether the osteoblasts do anything in particular to the cyclic F versus the cyclic F low neutrophils. So we show and see that the cyclic F neutrophils are inside the tumor nodule, but then in D and E, we ask, are they controlled by the osteoblasts? So we do 2 different experiments where we basically deplete osteoblasts and look at neutrophil production. And we see that we have fewer cyclic F high cells when we deplete osteoblasts. So it looks like...
But no change in the overall amount of neutrophils.
Right.
Or at least the cyclic F low doesn't actually change.
Right. So they don't change in frequency. They do go down in number a little bit, but like total number. Okay. But in E, where we look at the percentage of the hematopoietic cells that are produced, we see that there's no change in how many of the cyclic F low cells are produced. So it's really in the cyclic F high that we see the most striking difference.
Right.
So then... At that point, we said, well, okay, it looks like we figured out that we have 2 different types of neutrophils, so let's learn even more about them. And so figure 5 is basically all about profiling the cyclic F high versus low cells. And just to cut to the chase, we found that the cyclic F high cells had increased gene expression of a number of different genes associated with more tumor progression. So genes that are typically associated with more blood vessel formation or with direct effects on tumor proliferation and sort of increased myeloid cell recruitment. So these neutrophils express really high levels of CSF1, which is a gene that's really important for making more macrophages out of monocytes. So it was really they express all these genes that make them seem like they're more tumor promoting than the cyclic F low counterpart. And then we did a few other experiments looking, at their functions in vitro. So we looked at whether or not these cells actually could stimulate macrophage differentiation based on their, high expression of the CSF1. And that's what we show in D, that we have more macrophage-like cells in vitro when we culture them together with cyclic F high cells versus low. But I think the experiment that I particularly like the most in this figure is figure 5F. And the experiment we did here was we really wanted to know whether these different neutrophil populations had different effects on the tumor. And so we co-injected cyclic F high neutrophils with tumor cells into mice, or the cyclic F low cells with the tumor cells. And we asked, which neutrophil population, or do the neutrophil population change how that tumor grows? And we saw that if we isolated the cyclic F high cells and put them with tumor cells, the tumors actually grew faster. Mm. So that was, the main evidence for us that they actually had, tumor promoting properties.
Yeah. And it's quite impressive too. It's like not a small difference.
Yeah, I mean, it was...
And it's just better than CSF, which is like, I find that quite impressive.
So we wanted to know, it's not really enough for us to see, okay, they express these different genes, because that's, it's nice and interesting, but we wanted to know that they're actually functionally doing something to the tumor.
Right.
And most likely, all of these neutrophils are, a quite heterogeneous population of cells. So there may be some neutrophils that actually, kill tumor cells. There may be some neutrophils that actually promote tumor cells to grow. And so by splitting them up based on the cyclic F expression, we could see that the cyclic F expression basically enriched for cells that were more tumor promoting compared to the cyclic F low. And then I guess to end that figure, we wanted to go back to the human data and say, okay, now that we can distinguish these different populations, do we see a difference in sort of disease outcome if we separate patients based on their gene expression? So here gets a little bit tricky, because cyclic F is not actually a gene that's found in people. So we had to generate, a neutrophil gene signature. So basically, taking the cyclic F low neutrophils or high neutrophils, and then, taking a list of genes that they express and asking if we separate patients based on high expression of this gene signature, do we see a difference in survival? And we saw that lung cancer patients that expressed higher levels of the cyclic F high neutrophil gene signature actually survived a shorter amount of time than...
So here you're saying cyclic F high gene signature.
Yes.
So this is cyclic F is not actually in the human cell, but it's a gene signature from the mouse that you're translating into the human.
Yes, exactly. And now, we've gone further with these neutrophils in other studies in the lab, and we can see, on a, single cell level, basically equivalence between the cyclic F high cells in mouse and in a human. But it's even more granular than what we see in this paper. So, with every paper you learn something new, and then you learn, oh, it's more complicated, and then you go even deeper, right? So it's, this is basically a first approach to saying, okay, if we enrich for tumors or patients with tumors that have higher levels of these genes, and presumably then these cells, do we see a difference in the disease outcome?
Right.
And I think we'll just really breeze through figure 6, because basically this was our first attempt at trying to figure out, what is the tumor doing to act on the bone. And we identified one molecule that called rage, which was kind of fun because it was towards the end of my PhD, and I was like, yes, I'm feeling a lot of rage. I'm feeling this rage. So basically we could identify, we basically asked very broadly, if we look at serum from these tumor bearing mice and compare it with tumor free mice, can we find something that acts on the bone? And this is really like fishing, right? And it's really, presumably there's more than one factor, that it's complicated. But we were able to see that, okay, serum from the tumor-bearing mice actually increased osteoblast production in vitro. So if we did in vitro studies looking at osteoblast formation, we saw that, okay, serum from tumor-bearing mice had more osteoblasts. And then we did some detective work. We found that rage was increased in the serum. And then we did some more in vitro studies showing that, soluble rage basically was able to increase osteoblast formation in vitro, which was in line with some other published papers already.
Yeah, that's super cool.
Yeah.
So can I just ask a sort of more high-level question?
Yes, please.
Which is, why is all of this happening? Like this is super convincing work. It's really beautiful and sort of surprising, but very convincing. And so, but my question is do you have any idea or any speculation about what it is about the tumor that is sending a signal to the bone marrow to increase bone density? Because that by itself doesn't necessarily make any sense.
Right.
And then why are these neutrophils, which we normally think of as pro-inflammatory destructive cells, why are they somehow promoting tumor growth?
Mm-hmm.
Because you have to imagine two individual things. The tumor is signaling the bone marrow to alter the function of a couple of different types of cells, not only the osteoblasts, but also the neutrophils. And then those changes somehow are feeding back to promote the growth of the tumor in this distant site. So do you have any ideas about why that's happening?
Yeah. So
why either of those steps are happening?
So I would say the why question is definitely very difficult to answer in this case. But I think one of the things we haven't talked about so much is there's quite a large literature looking at how bone cells, without disease, in just a normal steady state, how they influence how immune cells are produced normally. So it's a lot of different groups have looked at, resident cells in the bone marrow and found that a lot of these cells are really important to provide structure, growth factors, chemokine gradients, different signals to immune cells that define which immune cells get produced and when they leave the bone marrow to go to these different sites. So it's kind of this idea that, these cells regulate hematopoiesis, basically. So what immune cells are made. And so you can imagine that in the context of cancer, like if you have, I don't think the effect on the bone is really directed. I don't think it necessarily benefits the tumor directly that the bone density is increased. But a side effect may be that, it's a kind of a feed-forward loop, right? That tumor acts on bone. That acting on the bone, in a mechanism that we still need to figure out, acts on, results in an increased production of these neutrophils that then funnel back to the tumor and help the tumor to grow. And you have more tumor, they make more whatever factors, including soluble RAGE, and then they funnel back to the bone marrow. And then, this goes on in a sort of a happy circle for the tumor. I think the tumor will just do whatever it can to grow, and it will use whatever mechanism that's already in place in the body and exploit those.
Yeah.
And it's not really, not invents new ways. You don't want to personify the tumor, but it will select for tumors that are able to use the mechanism that's already in place to make it grow faster.
Right.
So yeah, cool. I think there's lots to figure out still, and
oh yeah, for sure.
Lots.
The best kind of science.
Yeah.
Are you addressing anything remotely similar in your postdoc, or is it totally dissimilar?
So I basically now continued working in the lab to finish one paper, which is a single-cell RNA-seq investigation of the tumor microenvironment. And so we've looked more deeply into the lung tumor there and comparing sort of mouse and human and trying to see what are and specifically focusing on the myeloid cells. Mm-hmm. But in my future postdoc, which I will be starting in the fall, it's actually a neurobiology lab. But they have made this new technique called spatial transcriptomics, which basically allows you to get both spatial and transcriptional information about different cell types in a tissue.
Cool.
Yeah. So it'll be maybe tumor related, but maybe more neuro. Let's see.
Nice.
Yeah.
Well, that's neat. So just so our listeners understand, what this means is that we will try to be coordinating time between someone in Sweden.
That's right.
People on the East Coast, and then people in California. Well, one person in California.
So basically, I will have to stay up until midnight, and you will have a normal dinner time, and then Kate will have to go during work.
We'll be drinking beer for breakfast.
That's right.
We'll make it work.
Perfect. I mean, we will make it work, even if we had computer shutdowns and Audacity crashing and...
Yes. So listeners may have noticed that Chadene has not joined us for some time, and that's because her computer crashed again. It appears that both her audio files are saved, so I desperately hope
oh my
that
God,
is the
I hope
case.
so.
Because we are not recording this again. Camilla's paper appears to be cursed, but we will get this out come hell or high water. Thank you all for listening.
Thank you.
You can find our podcasts on iTunes or Stitcher or wherever you listen to podcasts. I don't think anyone listens to podcasts that is not subscribed to podcasts, but if you are one of those weird people that somehow finds random episodes of a podcast, like maybe all of Camilla's friends that haven't listened to the podcast up until this point, you should all go subscribe. Even if you're never going to listen to us again, just subscribe. It makes our numbers better. If you're one of the people that listens constantly, you should consider contributing to the production of the podcast by going to patreon.com/Audiommunity, where you can donate whatever amount you feel comfortable per episode. Episodes lately have only been coming once a month. Hopefully we would get more, but if you want to give us a quarter, give us $1 per episode. That's awesome. It helps motivate us to produce the show and also helps pay for things like web hosting and the audio software that I use to edit podcasts. We're also all over social media. You can find Camilla and I and Chadene and even Kate now on Twitter. There are links on our website, which is emunity.com or emunity.org, emunity.org, and the podcast is emunity.org/audiomunity. You can find us on Facebook, facebook.com/audiomunity. And what else is there? The music at the beginning and the end is performed by my wife, Rachel Reinick.
Fantastic.
Oh, and also we should say that Chadene has been writing these sort of paper summaries that are appropriate for people without immunology backgrounds. So if you want to share immunology with your friends, or if what we were saying is slightly incomprehensible and you want a different summary of it, you should go check out her website. And I'm just going to look it up. Hold on a sec. Psi-minded.
And it's a fantastic, the Psi-minded Idealist.
Yep. Oh, Idealist, right.
The Psi-minded Idealist, and she has one segment on lung tumors, talk to bone cells to promote tumor growth and spread. And this was actually before the podcast is even out because of our exciting
earlier snafu. Right. We'll put a link to the actual post in the show notes for this episode, which again you can find at emunity.org/audiomunity. And again, that's Audiommunity, not, as our hosts used to think, audio.
I think I just got that. Despite the
emunity.org, EMMUNITY.org. And I think that's it.
That's it.
We'll see you next time.
See you next time. Thanks for listening.